Assalamualaikum and hi, I am ASNAWI. So today we will cover chapter 3 in the biology subject, which is the cell division. So at the end of this lecture, you should be able to learn the concept of cell division, what is cell cycle, the process mitosis, and the process meiosis. First, let's take a look on the concept of cell division. Cell division is defined as the new cells that are being produced from old ones, and it involves two processes. The first process is karyokinesis, in which the cell divides and distributes its genetic hereditary material. This process is also known as the nuclear division. The second process is known as cytokinesis, in which the cytoplasm divides to separate the cells into two daughter cells. So towards the end of karyokinesis, cytokinesis will occur. This process can be shown in the figure, where in karyokinesis, the cells start to separate themselves, and moving towards karyokinesis, the cytoplasm divides to produce two daughter cells. Karyokinesis can be divided into two categories. The first category is mitosis, in which the nucleus divides to produce two daughter nuclei, which are identical to its parent. In other words, the daughter cell produced is completely identical to the parent cell and exists in diploid state. While in meiosis, the nucleus division occurs to produce four daughter nuclei with half chromosome set number. The daughter nuclei produced is in haploid state. So why do we undergo mitosis? Mitosis is needed for the growth of organisms, the healing or repair of damaged tissue, and even for asexual reproduction, or also known as the binary fission. And why we undergo meiosis? Meiosis is done for reproduction. So now we have covered the concept of cell division. Next, we will cover what is cell cycle. The cell cycle is defined as the sequence of stages that cells passes through one cell division to the next. The time taken to complete one cell division varies with tissue type and among species. So what is actually meant by the sequence of stages here? The sequence of stages here involves three stages. The first stage is interphase, followed by mitosis, and finally cytokinesis. This figure shows all the three stages involved in the cell cycle. The interphase, where the substages is the G1 phase, S phase, and G2 phase. The mitosis phase, where it can be further divided into four. And finally, the cytokinesis phase, where the cell finally divides to produce daughter cells. But now let's take a look at the interphase. In interphase, during G1 phase is also known as the gap 1 or growth 1 phase. During this phase, RNA and proteins are being synthesized. So when the RNA and proteins are being synthesized, the organelles will grow and increase to support the number of RNA and protein that are being synthesized in bulk. Since the number of organelles also grows and increase to support the protein synthesis, the cell also will increase in size to support the number of organelles that grow inside the cell. Therefore, in this stage, it will last for at least 10 hours due to the number of RNA, proteins, and other organelles that are involved in this phase. And then, after the G1 phase is finished, we will move to the S phase, or also known as the synthesis phase. In this phase, the DNA is being synthesized or duplicated, where each chromosome now consists of two chromatids joined at the centromere, and this process lasts for at least 8 hours. So this process is slightly shorter compared to G1 phase, but since the DNA sequence is long, it will need at least 8 hours, which is a long time for it to finish. So this is the chromosome structure. You can see here, the chromosome structure consists of two sister chromatids that are joined at the centromere region as pointed in the figure. And finally, we will move to the G2 phase. During G2 phase, the cell produces new proteins and continue to increase in size. The microtubules here, which is one of the proteins, are being synthesized for spindle fiber development, and centrosome is being duplicated, esters are formed and last for at least 5 hours. So in this phase, G2 phase is the shortest phase compared to the O3 phase. This is so because all of the organelles and proteins have been synthesized, and now it only focuses on the synthesis of microtubule for spindle fiber development. And finally, the cell will be ready to proceed to the cell division. So we have covered the concept of cell division and the cell cycle. Now we will move to the mitosis process. So after interphase, the cell will undergo cell division where it starts with the mitosis. Mitosis can be divided into 4 phases as being shown here. Mitosis is defined in which the chromosomes only are being replicated and divided to obtain 2 new cells that are identical to the original cell. This process is followed by cytokinesis. Mitosis can be divided into prophase, metaphase, anaphase, and telophase. So first, the mitosis always starts after interphase. After interphase, cells are already prepared for keratin. Here we will start with mitosis. Mitosis starts with prophase. During prophase, the chromatin condenses into thick coils to form chromosome. The chromosome form consists of sister chromatids joined together with centromere. The nucleolus will disappear, the nuclear envelope will start to break down, the centrosome and centriole will start to move to the opposite side of the nucleus. Then we will move to the metaphase. During metaphase stage, the chromosome will be lined up at the metaphase plate attached to the spindle fiber. And moving on to the anaphase, the sister chromatids will be moved towards the opposite side of the poles through the shortening of the spindle fiber. And finally, in telophase, the chromosome will start to uncoil and returns to the chromatin form, the nucleolus reappears, the nuclear envelope reforms around each set of chromosomes. Now, after telophase, the mitosis will proceed with cytokinesis to produce 2 new daughter cells that are identical to the parent cell. However, animal cell and plant cell will undergo different kinds of cytokinesis. As for animal cell, a cleavage flora will be formed, where the flora will deepen inwards until it reaches the center of the cell, dividing the cell into 2 daughter cells. While in plant cell, first the vesicles will form at the equator of the dividing cell. The vesicles will enlarge and fuse to form a cell plate. Finally, the cellulose will be laid down between the new membranes, forming a new cell wall between the membranes, and finally produce the 2 new daughter cells. So now we have covered the concept of mitosis, and finally we will move to the process of meiosis. So by definition, meiosis is defined as the process of nuclear division in a cell by which the chromosome number is reduced to half, from the diplete number to the half-plate number. The meiosis 1 reduced the number of chromosomes by half, while in meiosis 2, the double-stranded chromosome is being divided to form a single-stranded chromosome, or also known as the chromatids. This maintains the diplete chromosome number across generations, while increases the genetic variation when male and female gametes fuse together. So just like mitosis, meiosis also starts after the interphase stage. During interphase, the cell also has been prepared to undergo meiosis, and meiosis will start with the prophase 1. In meiosis 1, during prophase 1, there's a number of interesting events that are occurred here. The first will be the chromatin will start to condense into thick coils forming chromosome, the nucleolus will disappear and the nuclear envelope will break down, centrosome and centriole will move to the opposite side of nucleus, which is just the same with the metaprophase in mitosis. However, for the chromosome, the homologous chromosome will start to pair themselves, where this process is known as synapsis, to form bivalents or tetrads. Here, the crossing over will occur at plasmata to exchange the genetic material between the homologous chromosomes. The prophase 1 can be further divided into 5 different substages, where the first stage is the lactotine, where the chromosome starts to condense. And then we move to zygotine, in which the synapsis between the homologous chromosome occurs, and moving on to the fecitine, where the crossing over will occur at plasmata, and then the diplotine, where the synapsis occurs starts to separate due to the dissolution of synaptonymal complex. The synaptonymal complex here is the protein that assists in the synapsis process. And finally, the diakinesis, in which the nucleus and the nuclear envelope will start to disappear and breaks down respectively. And then we will move on to the metaphase 1. In metaphase 1, instead of the chromosome, the homologous chromosome will be aligned at the metaphase plate. And during the anaphase 1, the homologous chromosome will start to separate and begin migrating towards the opposite pole, pulled by the spindle fiber. Compared to the anaphase and mitosis, where only the sister chromatids move, in anaphase 1, the homologous chromosome separates and moves to the opposite side of the pole. And finally, we will move to the telophase 1 and cytokinesis. The chromosome will start to be condensed, the nucleolus reappears, the nuclear envelope reforms. After telophase 1, each half of the cell has a complete haploid chromosome set. One or both chromatids include regions of non-sister chromatic DNA. Remember, during prophase 1, there is a crossing over occur that allows the exchange of genetic material between two non-sister chromatids. This causes one or both chromatids will include the regions of non-sister chromatic DNA during telophase 1. So after telophase 1, the cell will produce two new daughter cells with half of the chromosome number. And finally, the meiosis 2 starts with the prophase 2, metaphase 2, anaphase 2, and telophase 2. The process here is similar with the mitosis process that we have looked through earlier. But during meiosis 2, the number of daughter cells produced is 4 compared to mitosis. So here is the comparison between mitosis and meiosis. Here we can see for mitosis, only the chromosome is being duplicated, but for meiosis, a number of interesting events occur, such as synapsis and crossing over. And during metaphase, in mitosis, the chromosome is being aligned at the metaphase plate. However, during meiosis, the homologous chromosome is being aligned at the metaphase plate. And anaphase, proceeding to anaphase and telophase, the mitosis process will finally produce two daughter cells which are identical to the parent cell. However, for meiosis, during telophase 1, it will produce two new daughter cells that has half the chromosome set number compared to the parent cell. And during meiosis 2, in telophase 2, it will produce four daughter cells that have half of the chromosome number compared to the parent cell when they undergo further division in meiosis 2. So in summary, for DNA replication, in mitosis, DNA replication occurs during interphase before mitosis begins. However, for meiosis, the DNA replication occurs during interphase before meiosis 1. Mitosis will undergo one number of division, and meiosis will undergo two number of division. In mitosis, there are no interesting events occur, but for meiosis, synapsis occur during prophase 1 along with crossing over between non-sister chromatids, which results in plasmata that all pairs together due to sister chromatid cohesion. For mitosis will produce two daughter cells, each will be diploid and genetically identical to the parent cell, while for meiosis, it will produce four daughter cells, each in haploid state, which contains half as many chromosomes as the parent cell, which are genetically different from the parent cell and from each other. The role of mitosis in animal body is to growth, repair, healing, and in some